Hello everybody, my name is Lammert Albers with the Global Alzheimer's Platform Foundation. During the month of April in 2022, the FDA issued draft guidance for improving enrollment of participants from underrepresented racial and ethnic populations in clinical trials. Now with CTAD, one of the most consequential meetings on the calendar in AD drug development, and it being around the corner less than 4 weeks from now, we reflect today on how diversity will transform our understanding of Alzheimer's disease and trial design. Bio-Hermes is a landmark biomarker study that investigates blood-based and digital biomarkers in diverse participants screened for Alzheimer's disease. I'm joined today by John Dwyer, President of the Global Alzheimer's Platform and sponsor of the Bio-Hermes trial. Also with me today is Wolz, Chief Science Officer with IXICO, a neuroimaging company. Dr. Wolz is presenting the Bio-Hermes data during the opening symposium at CTAD, the effects of race and gender on amyloid positivity. Now, John, can you start by providing some context on the Bio-Hermes study and the aims of its study?
Sure. So when we started this study, we had two very ambitious aims. The first was we saw the explosion of need that was going to occur with the approval of disease-modifying therapy, and this is back in 2020. And we knew that the healthcare system and the clinical trial system needed to get much more sophisticated with the way in which it conducted its processes to provide better assessments and diagnoses of disease with higher reliability, more sensitivity and specificity. There are a number of emerging technologies that purported, and we were optimistic they were being accurate, that they could vastly contribute to these needs. But the Bio-Hermes study is designed to actually validate their technical performance in relation to the gold standard that Dr. Wolz is in command of, which is beta amyloid PET. One of only two methods that the FDA has approved as a diagnostic for the presence of beta amyloid associated with Alzheimer's disease. So we essentially created a side-by-side comparison biomarker study, and we wanted to see the results of what would happen when we screened 1,000 people in this endeavor. We'll talk more about that in a second. The second was we wanted to be sure those 1,000 people look like the people that get Alzheimer's disease. And in that regard, we made a very aggressive commitment that the trial would be composed of a minimum of 20% non-white Hispanic people. And we ended up with 11% African American and 11% Hispanic origin in the study. So a total of 22% is a terrific result for us and the field.
That's great. So Robin, why does the industry need to increase focus on diversity in AD clinical trials?
So first of all, the need is there, of course. As we're developing treatment, we need to make sure we're validating that in a representative population, and that's reflected in the FDA guidance you've mentioned in the beginning. The problem we have when it comes to biomarker research is lack of data. So for example, if we look at some of the late stage treatment trials in those drugs that were recently approved, like aducanumab, where we had less than 1% of participants were African American and less than 2% were Hispanic. And we have a similar situation in natural history studies like ADNI, for example, the Alzheimer's Disease Neuroimaging Initiative, where a lot of the research on those biomarkers is based on that it is not representative of the overall population. And therefore, there are open questions around the performance of different biomarkers in those different populations. For example, when we think about amyloid PET, which is, as John described, one of the gold standard measurements when it comes to amyloid pathology, that is obviously interpreted in a binary way when it comes to diagnosis or, in this particular application, clinical trial inclusion. Now, because of the limited data available in diverse populations, there are open questions on how this biomarker is comparable across different ethnic and racial groups, for example. There is limited data emerging now, and that shows that there are potential questions on whether those cutoffs or those interpretations of the measurements that we've established in the predominantly white populations that most data is collected in equally apply across all populations. And to answer those questions in more detail, studies like Bio-Hermes are critical. So to get data rich in biomarkers in those populations, and then try and interpret that data to understand whether the learnings that were established in the data we have equally applies across those populations.
That's great. So, gentlemen, how do you feel diversity and imaging biomarkers will help shape our understanding in clinical development in general?
Dr. Wolz is the scientist, and so I'm going to defer to him on the science part. But looking at it as a patient advocate and as from the point of view of a family that's been stricken many times by the disease, we owe it to patients to put their mind at ease that therapies work for everybody. We did not design Bio-Hermes with the expectation that the results would be different for African Americans or Hispanics. We just don't know. And as a consequence, we need the data to be able to shed light on whether there are or not differences that are material by race or ethnicity or gender, and to create real rigor in that analysis. But I'm hopeful that we're going to find important explanations for potential variations, but we need the data, and without the data, we're being unfair to the field.
Yeah, and maybe one aspect just to add to that is one key reason why we need more data and why we need to understand those relationships better is because we need to, so to speak, disentangle any potential differences in the biomarkers from correlating factors like socioeconomic status or comorbidities, for example, that we may see between those different racial and ethnic groups. For example, some studies have been showing an increase in cerebrovascular disease in Black populations compared to white populations. And if we keep that aspect in mind, then that could potentially explain why the biomarkers from an amyloid PET, for example, are not comparable in subjects that have the same level of clinical dementia. In other words, whether there is a comorbidity that is more pronounced in one population than in the other, that makes interpretation of those biomarkers more general. So this is a very, all of these questions are intertwined and interconnected, and therefore it is important to get the data to understand how those different questions link together, so we can ultimately make sure that we can use that information to design more specific inclusion criteria for clinical trials, improve recruitment rates in those underrepresented populations, and ultimately increase chances of getting successful treatment to patients.
Right. That's great. So finally, as we seek to wrap up this episode, what do you feel is the aspirational impact on clinical trial design in Alzheimer's disease?
So for GAP, Lammert, I think the the things we've just briefly discussed here, when we talk about race and ethnicity and we talk about biomarkers, we're creeping up on really talking about precision medicine and the ability, because I don't want to sound like Dr. Wolz here. He's a scientist, but a self-reported status, like I'm a African American or I'm an Irish Catholic, are social classifications. They aren't clinical classifications. And we've got to get behind the data and understand what are the unique genetic, as one example, or the unique biomarker configurations that may make you more susceptible or less susceptible to demonstrating beta amyloid accumulation than the majority of people, and start to sort at a much more individual level how people are going to react to medications and how they are going to express the disease. The work that we've done with IXICO is the first step in getting more rigorous and more insight into genuine underlying physiological and clinical characteristics to help us sort the right diagnostic process and the right therapeutic process.
John, Robin, thank you. As a reminder, Dr. Wolz will be presenting the Bio-Hermes data during the opening symposium at CTAD. Once it's sold out for in-person, you can still register for online participation. The symposium will take place Tuesday, 24th of October. It will take place in the Grand Ballroom at the Boston Park Plaza at 4:55 p.m. Eastern Standard Time. And it's the effects of race and gender on amyloid positivity. The chair of the session will be Susan Schindler, and she's from WashU University in St. Louis. Thank you.